I was muted. So our today's lecture is based on pharmacology, autonomic drugs. Okay, and yesterday we studied the in pharmacology lecture. Yesterday we have studied only the first page of we only studied this. Okay, but in yesterday's physiology lecture, neurophysiology lecture, I told you everything about the acetylcholine receptors, all the neurotransmitter receptors, even epinephrine, norepinephrine, and also I explained you the maturation control. I explained you the alpha, beta, all 5 types of M receptors, the nicotinic receptors, the neuromuscular junction. I have explained all these things, okay, in last previous classes. Also, the second messenger system I have explained you. In yesterday's neurophysiology lecture, we have covered the whole location and the function of all these receptors, and also the second messenger system. We have studied the GS, GI, and GQ system in a lot more detail. That is not given in that detail in this first aid book, but I have taught you in very much detail, so the people who have like missed the yesterday's live class can check your drives for yesterday's neurophysiology lecture. Okay, so you will get all these details of this. So now today's lecture will be based on the autonomic drugs. Okay, we will start from autonomic drugs, and before going to that, I actually don't like this little picture that much than the picture of Lippincott. So kind of pause sharing, and I will open up the Lippincott picture for you so that you may grab it more better, because that's the basic textbook, right? So resume share. Okay. So people, let's begin with the synthesis of acetylcholine. Okay. Now, we all know that we have this acetylcholine wrapped up into a synaptic vesicle, or we call it as a storage vesicle. Okay, but we will start it from the beginning. Okay, now there is this for example, this is the neuron. This is inside the axon and the dendrite, and then this particular level, we have the extracellular fluid, okay, the ECF. So the choline, which when the acetylcholine at this level, I am so sorry, wait a second, let me open it in another tab so that it may not get closed. Okay, here people, you see in the synaptic cleft, when acetylcholine is released, it is destroyed into two via an enzyme called acetylcholine esterase. It is also written in this box. Okay, so this acetylcholine is destroyed. It is hydrolyzed into its two components, that is acetate and choline. Okay, so this choline goes up back again into the extracellular fluid, and then it enters into here again. So this is the first step. Here we have the sodium transporters that along when the deep when this neuron has a depolarization signal. Okay, and I've told you that previously in the neurophysiology lecture that they works on the principle of sodium potassium ligand gated channels. Okay, so we have this sodium channel on the neuron also. Okay, so in this presynaptic neuron, along with sodium, the choline enters, and then they both, when they this acetyl coenzyme A joins at this particular level, okay, there is a reaction occurs because of this enzyme, which was not written in the book, so I've kind of written it over here, choline acetyl transferase. Okay, it transfer this acetyl coenzyme A. From this acetyl coenzyme A, it transfers the acetyl with the choline, they combine up, and then it forms this acetylcholine neurotransmitter, which is an ester. Okay, now at this particular level, this acetylcholine is wrapped up into these vesicles, okay, into these vesicles, and then for these vesicles to get you know touched at this presynaptic membrane, when they come and strike the membrane, then the vesicle disrupts and acetylcholine goes out into the synaptic cleft. But this process is not that so simple. Even in this diagram, there are so many things that are not mentioned. For example, let me tell you, if you want to write, you can. There are certain proteins present over here, okay, that helps tie up this vesicle to the wall. Let me draw it. Okay, you know just take it like that. A protein holds this, another one of them binds it with this wall, okay, another one push it down to here, okay, so there are three. And then when it comes down, suppose this is a whole full membrane, it comes down, it strikes this membrane, then the exocytosis process occurs. So there are proteins that are involved in these things. Okay, if you want to write the name of protein, they are synaptobrevin. Okay, they are synapto. This will also help you in understanding the cell physiology. Okay, so you can write it, maybe you can see it somewhere. Synaptobrevin. Okay, one of them is this one, and the other important one is syntaxin. Okay, syntaxin. Forget about my writing. It's very hard to write on a keyboard. So synaptobrevin and syntaxin, these are the two proteins that help bind these vesicles to this wall. Okay, so this all process occurs by the influx of calcium. So calcium is very important for the depolarization mechanism. Okay, because of this calcium, the synaptic vesicles pops out, and then this acetylcholine goes into, and this whole process happens. So now, just for instance, this acetylcholine is now into this synaptic cleft. Okay, this region is called synaptic cleft, and this whole thing means that the presynaptic neuron, the postsynaptic neuron, and this space, this whole thing is called as a synapse. Okay, so never confuse in between a synapse and a synaptic cleft. Cleft is only a place, a space, a hollow portion that is connecting two neurons, presynaptic and postsynaptic. Okay, so now you see this acetylcholine is released into this space, and then this acetylcholine is kind of um, attaching to the membrane. When some of that goes and attached binds to this receptor. Now this receptor can be M, okay, any of the M, uh, based on the location where it is present, okay, any of the muscarinic one, or it can be nicotinic one, because we have only two types of cholinergic receptors that we studied yesterday, muscarinic and nicotinic. So this acetylcholine can bind to any of these two receptors, okay, and the rest of the acetylcholine that is left over here will go back and attach again to this presynaptic receptor, because it's a presynaptic neuron, so this is a presynaptic receptor, and the name of this receptor is alpha 2. Okay, in all neurons, alpha 2 is present at the presynaptic terminals. Okay, so it goes here and it attaches over here, and then this receptor gives the signal back up, okay, back up to here at this particular level, that please do not produce or make more acetylcholine. We do not want it. We do not want this. So this is essential to understand. Okay. Now, on the basis of this mechanism, like this is the basic mechanism of acetylcholine, okay? And this is how it works. Now, I want to tell you one more very important thing that you might read in books or in first aid. It is not like that much given, but like in exact terminologies, but there is something which I want to tell you is called cholinergic crisis, okay? We have cholinergic crisis. What is cholinergic crisis? When this acetylcholine is left over too much in the synaptic cleft, okay, then the signals kind of goes up, and then they give the signal that we do not want it more. So before this thing happens, when this is left in more here, there is this enzyme which is breaking this acetylcholine into choline and acetate, okay, which is hydrolyzing the acetylcholine esterase enzyme, okay. It inhibits. This enzyme is inhibited. When we do not have this enzyme, okay, there are certain conditions when these enzymes are inhibited. So when this enzyme is inhibited, this acetylcholine is not break down into this tube, this process stops, and there is a lot more concentration of acetylcholine left in the synaptic cleft. This condition is called cholinergic crisis, okay? So the patient undergoes the enhanced cholinergic effects. And to study the enhanced cholinergic effects, we will go back to the first aid. And you have to see this part, okay, that we have this cholinergic. Is the lecture recording? Yes. Okay. Now, M1, M2, M3. Okay. So as I told you yesterday, the locations of these cholinergic, but the best symptoms you will see in cholinergic crisis, or you can say the best adverse effects. You know, the adverse effects of the cholinergic drugs, the agonists, the adverse effects of them are basically the actions. For example, just let's take an example of a drug, bithanicol. Okay, that's the first drug we will study. So this drug, bithanicol, has increased effect on the bladder, on the bowel. Okay, so I remember it as 3Bs. So you see, on the bladder, we have M3. On the bowel, we have M3. Okay, so this drug is causing effect on bladder and bowel, but M3 receptors are present on other areas as well. They are present in the lacrimal glands, the sweat glands, the salivary, the gastric parietal cells, okay, that secretes HCl, which we called hydrochloric acid, the gastric acid, okay. So gastric parietal cells, then in the gut smooth muscles, which are involved in the gut peristalsis movement, that is, the food is only gone forward but not backward. Okay, that is what we call peristaltic movement. The food can only go in a forward direction but not backward direction. Then in the bladder detrusor muscle, okay, the bronchus, the bronchus smooth muscles, the pupillary sphincter muscles, okay, I told you yesterday, levator palpebrae, okay, the radial muscles of the iris, all at all these regions, the ciliary muscles, okay. Then in the pancreas, in the endothelium of the blood vessel, the endothelium of the blood vessel. So you see, this is the location of all these uh, M3 receptors that is present in all of these locations. So when we are taking a drug, it is not going and acting on all the M3. No, 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 no. That is not the thing how it works. That is the beauty of drugs. This drug bethanical will only work on the bladder, okay, and on the bowel. That's it. No other functions you will see. But when this drug is, like, you give an overdose of this drug to the patient and that drug is left over in this synaptic cleft more, okay, what this drug will do, you are not having this sufficient acetylcholine, you're not having, there is some problem at this particular level. Maybe the problem is because of hemicolonium, okay, which is responsible for the transport, okay, and now sorry, not responsible, which inhibits the transport of choline, okay. It does not allow this process to occur. This hemicolonium inhibits this step, and acetylcholine esterase does not work. This acetyl coenzyme A does not added up with choline and makes acetylcholine. So this step is inhibited. So maybe there is problem at this level. Maybe there is problem at this level, that they are not you know, binded up into this vesicle. Maybe there is more degradation of this vesicle. Maybe there is problem in the uptake. Maybe there is problem in the synthesis, or maybe it can be a problem in you know, the release. There is another toxin drug, or not a drug, basically it's not a drug, it's a toxin which we call botulinum toxin. It is present in foods like mushrooms, etc. Okay, so if this botulinum toxin, the patient got this in the blood, okay, so it will not let those synaptobrevin or syntaxin, which will help bind this vesicle to this membrane, it will not bind up in here. So you see, there can be a problem in release as well. There can be a problem in synthesis, in uptake, in release. Okay, so there is another thing called spider venome, which is if a patient got bitten by a black spider, black widow spider, we call it. So in the poison, the venom, if gets into the blood, it can also hinder this process. So it can be any of the reasons. There might be a problem in acetylcholinesterase. For example, in the disease called myasthenia gravis, there is a problem in here. Okay, so you see, there can be any problem. There can be a problem in firing of calcium. So no stimulus, no movement of the synaptic vesicle towards the membrane, no process. So it can be a problem anywhere. So in those cases, when the patient do not show these kind of signals, okay, then or the patient have lots of acetylcholine in here, and you know, the patient is showing adverse effects of acetylcholine. In those cases, we give the patient these kind of drugs depending on the condition, that either we have to give them agonist or either we have to give them antagonist. or either we have to give them the direct agonist or the indirect agonist. So all of these, which we will study from first aid next, okay? So right now, I just want you to make this concept better, that just say, for example, this whole thing is blocked and we have this betanicol here. This is betanicol, okay? Now this betanicol is a direct agonist. It will bind in here directly. And what will it do? According to its duration of action, okay, it will act. After some time, when its duration of action is finished, it will be hydrolyzed. It will be finished. If you do not have a good concept of zero order and first order kinetics, I will definitely touch that portion as well. Okay, so do tell me in the WhatsApp group, because that is very important to understand how a drug is hydrolyzed. Okay, so because there are drugs that work on the mechanism of zero order kinetics or on first order kinetics. Okay, the process of decreasing its concentration into the plasma. Okay, all that thing. So you see, for now, botanicol is just attached over here, and and this is an M3 receptor, because it works on M3 receptors only. But this M3 receptor is present in all over this area, okay? But its function is on gut and bladder. This is the beauty of drugs. It does not cause problems to all of them. It only acts in the region for where it is meant to be, okay? So this is one thing that I want to tell you. Another thing is that in case of adverse effects, okay, all these actions, these are all actions, okay, of acetylcholine. These are all actions of acetylcholine and the drugs that are acetylcholine agonist, okay? But when I talk about the adverse effects, when at this particular level the concentrations of these drugs are very high, or when the concentration of acetylcholine itself is very high because there is no acetylcholine esterase. In such condition, when the patient undergoes cholinergic crisis, the adverse effects that are seen are exactly the same. These are the adverse effects. So I am kind of trying to make you understand this concept, that whenever you will study the adverse effect of cholinergic drugs, you will see excessive sweating, excessive salivation, increased gut mobility, or excessive urination, because when the bladder will contract more, the patient will urine more, okay? And like more and more, accommodation and, meiosis, all these things. Decreased level of glucose, because when the insulin is released more, the glucose is decreased, okay? Excessive sweating also because of this vasodilation process, thing, excessive lacrimation, more and more tear production. So all of these actions will become side effects or adverse effects in case the patient undergoes any sort of cholinergic crisis, either due to physiology, like physiological problem with acetylcholine esterase enzyme, that acetylcholine levels are more, or because of any drug. The adverse effects will be the same as actions. So just keep this thing in mind. Now we will move to the drug portion, okay? So I am right now only touching this little part, okay? And I will, if you want to kind of revise a bit about this Q and I. So yesterday I told you that we have this GS, GQ, and GI systems, okay? And the GQ system was the one that works with phospholipase C, okay? It activates the phospholipase C, and then the end product was always protein kinase C, okay, and there is an increase in this calcium, intracellular calcium, and IP3 inositol triphosphate and DAG mechanisms. So from the sarcoplasmic reticulum, more and more calcium is released. So now you get this mechanism of GQ, that when you have more and more calcium coming out from sarcoplasmic reticulum, that calcium will stimulate this process. So this is important. That is why this muscarinic receptors actually work on the basis of they work on the basis of this this GQ system. Okay, then we have this I, the inhibitory one, that inhibits adenylyl cyclase, and that adenylyl cyclase will not let ATP to convert into cyclic AMP, and cyclic AMP is further responsible for protein kinase A activity that will help in phosphorylation and opening of sodium potassium ligand gated channels. So all that process is inhibited, and so does the heart rate and contractility of atria. Also, you might, if you remember, I told you repeatedly again and again that when we talk about the parasympathetic, or we talk about this M2, because this decreased response in heart is always parasympathetic, right? The increase is sympathetic. So we have, when we talk about the myocardium of atrias, that is a parasympathetic effect. And whenever we talk about the myocardium of ventricles, then the myocardium of ventricles have more sympathetic effect. Okay, so that is why, see here also it is written. So these are very little words which when we read this, we forget putting focus on it, and then in exams we miss the MCQs, and when we like kind of come back home and search it in books, and was like, oh my god, it was written over here. So these are the mistakes we usually make. Okay. Now I will go to, so I will touch this alpha part again as I touch this cholinergic part when we will be studying the adrenergic drugs. Okay, so the first drugs in our first aid are basically the autonomic. So I have explained you this acetylcholine mechanism, and I will explain you this norepinephrine and epinephrine mechanism, the adrenergic one, okay, when we will be studying the adrenergic drug, so that you may not mix these two. Okay, so for now you only have to remember this. So let's see what this picture has to tell us. It is the same story. The choline is coming from in here, what was degraded, it goes back into the extracellular fluid, and then it enters in here via sodium channels. Now you see the sodium is not here, so that is why I didn't show you this picture. It is not like good and complete. So here at this level, we have this enzyme which I told you that was acetylcholine choline acetyl transferase. So okay, so that's what they have written over here as in CH for choline, A for acetyl, and T for transferase. So a chat, okay, and then we have this acetylcholine. Now they have not mentioned in over here. If you are like having your book with you right now, you can just at this step you can write that this step can be inhibited by hemicolinium, okay, as it is it is written in this picture of Lippincott book. Okay, this is a picture of Lippincott book. So it is inhibited by hemicolinium. So you can kind of write it in your book in here at this level. Okay, then you see it is being packed up in this synaptic vesicle, and then again this calcium is coming in, and further on the process is going on. But this is important at this particular level. This is very important at this particular level, that we have this botulinum toxin. Okay, we have this botulinum toxin that is inhibiting this particular step. So if it is given in first aid, it can be asked in your exams as well. So you can kind of mark this point, that botulinum toxin hinders the synaptic vesicle attachment to this presynaptic membrane. Okay, and you can see in here also. So basically it inhibits the release of the neurotransmitter, the botulinum toxin. Okay, then um, then again, this all whole process, acetylcholine esterase, this green color thing is acetylcholine esterase, which is present on the membrane of postsynaptic neuron, okay, postsynaptic membrane, and it will break this acetyl and choline into two, and choline will again go back up. So you understand this picture really well now, okay? So this picture I will explain you when we will start the adrenergic drugs, okay? And all this above is written about this noradrenergic thing, so that is why. Okay, so for students, it is like nine minutes left to end this 40 minutes class. So I kind of if we you know, if there is any problem, if the class ends, we will continue the class, because we have to cover our drugs today, at least the direct agonist for these. At least I will cover these four drugs today, okay, in detail. So you have to come back again to have this lecture continued, and I will wait for you people. So do come quickly. So it's 9 minutes still remaining, so we will kind of continue, okay? And the link will be the same. Just tap on the same link that has been sent to you on the WhatsApp group, okay? Now you see this drug which I was talking about, these are the direct agonists. So direct drugs are those which directly bind to the receptors, okay, which will directly bind to this receptor at the postsynaptic membrane. This though the drugs which directly bind are called direct, and the drug which are called indirect, that basically works on this enzyme. The indirect drugs do not bind directly on it, but what they do, they inhibit this enzyme activity, and then they come and work. So those drugs do not bind on the receptors, they let acetylcholine work actually, you know, they let acetylcholine work actually, and they inhibit this enzyme so that acetylcholine may remain at that particular place, the synaptic cleft, more, and can show its enhanced actions. So that is the difference between a direct and an indirect drug, okay? So we will be studying botanical right now, and you see, you can remember this with 3Ds, so it will be easier for you to remember. Also, let me tell you, the duration of action of this drug is 1 hour, okay? It is like, it works for 1 hour. That is the duration of action for this particular drug. And then I kind of have to remember its receptor, which will be M3, okay? So you can write it down over here in your book, that it works on M3 receptors, because M3 receptors are present in the bladder and the gut, okay? And now what they're doing, it activates the bladder smooth muscle. That means the urinary bladder will this the trusor muscle of the bladder will feel pressure, okay, and the patient can urinate, okay? So it does that. So it increases the micturition, okay, by relaxing relaxing the retrusor muscle, not by relaxing, by contracting, okay? By contracting the retrusor muscle, the patient gets the feeling of micturition. So that is its function. Okay, then it is resistant to acetylcholine esterase. That is, if this drug is present at this particular level, if this drug is present at here, it is binded up in here, it reaches here, okay, some of it is binded over here. This acetylcholine comes very happily, that I'm gonna do my work, and I'm gonna destroy this just now. But when it comes and do this action, there is a protective shield. This drug is resistant to this enzyme, so this enzyme cannot work on this drug, Okay, so that is why it performs its actions. Then no nicotinic activity. So it does not work on the nicotinic receptors. It does not work on the nicotinic receptors. It only works on the muscarinic receptors. Okay, and then the therapeutic applications, that if you are working in a surgery unit, you're a house officer and you're working in a surgery unit, and there comes a patient who is complaining of bladder outlet obstruction. Okay, this is a word which is used clinically, bladder outlet obstruction, BOO. Okay, what happens is the urine is not coming out. Okay, it is stored in the bladder. That is outlet obstruction, not coming out. So that terminology is written as diagnosis. Okay, the complaint of the patient will be urinary retention, that you will write that the patient is unable to pass the urine, that is urinary retention, but the diagnosis will be bladder outlet obstruction. Okay, so in that particular condition you can use this drug. So this drug is really good because you know that it will help the patient work in 1 hour. Okay, and also it will not affect the other organs. The patient will not complain of excessive sweating or salivation. Yes, there is an adverse effect of this drug, all of them. Like, it is not written here, so like I'm kind of telling you that the patient will undergo sweating, will undergo salivation, excessive tearing, hypotension because of this vasodilation. Okay, which we just studied here. Because of this endothelium mediated vasodilation, the patient will undergo hypotension, the patient will undergo bronchoconstriction, the patient will have diarrhea because of the increased GI motility. Okay, the patient will have sweating, or because of this vasodilation the patient will have flushing. Flushing is redness of cheeks. Okay, your cheeks become red. So that is what we call flushing. So the patient will undergo flushing, the patient will undergo vasodilation. So all these effects will be seen in that person as adverse effect of this drug. But but only when the quantity is more. Okay, when the quantity is too much. But you see, in every drug you take, it has some side effects, but it does not show all of them in every individual. So that's the case. So in your exams, all kind of side effects are not important. Only those side effects are important that cause serious threat to life. Okay, and similarly, all applications are not important. For example, it is working on GI also, but we are using this drug more in bladder outlet obstruction, urinary retention. So you kind of remember it, or they can in like paper 1 it is very straightforward, but paper 2 is not. So you might can get a scenario, for example, like the patient undergoes surgery. Okay, and after the surgery the patient has this urinary retention, or there is a pregnant female who just gave delivery and she's in her postpartum phase and she's having this postpartum urinary retention. So the drug of choice will be buthanicol. Okay, so you will only give this drug after a scan. Okay, whenever a patient comes with this type of problem, you can suspect there might be a stone in the bladder. The urethra is the stone is stuck in the urethra, and that can be a problem for urinary retention. So whenever, before giving a drug to the patient, you have to make sure that you do this ultrasound KUB, which is called urinary bladder. Okay, and also it involves the urethra because it's very short. So this ultrasound is important to rule out any stones or obstructions anywhere, and then only you can proceed with the treatment. Okay, so these are some clinical points that I'm telling you right now. Also, uh, uh, not only postpartum or not only, you know, surgical problems in that cases. You can also give this if there is some neurological problem, which you call as neurogenic atony. Atony is means no tone. The muscles are having no tone. They are feeling no pressure. So in, if the nerves are having problems, the nerves that are coming at this particular level, they are having problems. So that what we call neurogenic atony, and the patient is not able to feel the feeling of micturition. In that particular case, you can give with honeycomb. Okay. And also you can kind of remember, if you want to know about its effect when it's given in GI. It is not mentioned in your book, but you can write it down. It increases the GI motility. So there is a condition in children, mostly it is in children. uh, you have heard megacolon. Okay. What happens in megacolon is there is a set of nerves, a ganglion present